So today, I'm going to show you my main PhD results, okay? So I try to make very simple, and so you can take a message to home to share with the family, okay? So, yes, so my supervisor was Rita Henderson, and so the photo here, there. Okay, so today, I'm going to talk about flux. And thanks to David, he talked about flocculation and coagulation, and this is what was my PhD, making flux. So, but before I introduce what is flux, I'll show you what we do in this. Okay, we know algae is a pain, cyanobacteria is a pain. For catchment problems and everything, and in the treatment plan is also a pain. Because, you know, cyanobacteria the concentration can rise very, very, very quick. It's very difficult to control. And plus, different from organic matter, we got algal organic matter. So here, there are cells. So they are like a human beings. They excrete things. And that make more challenge in the treatment plan. Okay? So what it is people are familiar with this. Just very simple way of what is coagulation and flocculation, okay? So we got the cells are negative charge, so they don't like each other. So you have to put something to agglomerate them, okay? So this something is a chemical called coagulant, okay? So you got the cells in the tank, you put the coagulant, and you make the flux. So this is the flux, this is why I started doing my PhD, flux. Okay, when you have the flux with the bigger particles, they are very easy to remove in the treatment process. You can use air, so you can do dissolved air flotation, and just put the flux on the top of the tank, or you can remove by sedimentation. But when I said before, cyanobacteria is a pain, so you can have a clean water and things like that. But the thing is, when you force flux, they can break, okay? And they can go to your filtration process, they can go to your chlorine tanks, and can give you something like that, okay? So like it's a little bit worse scenario, but can give you color and taste and other problems, no? And all, what you learned this today is all the toxin and all these kind of nasty things they can release, okay? So this is what we start in flux, because are very complex, and also you need to something new when you're doing a PhD. Okay, so the thing is about we has a methodology how to characterize the flux. Okay, so the aim of this study is just to show how different cells, cyanobacterial cells, make different flux properties, and what is the challenge in the water treatment plants. Okay, how we can understand flow properties, and how can be done these flow properties how can be improved in the downstream processes in the water treatment plant. Okay, so I started 4 species plus 1, but this is green algae, so I didn't include that one. But we got 2 microcystis, 564, 555, and cylindros, and alias Anavena, they changed the name, but it's still Anavena for me. So anyway, it's 4 species. Okay, and this is a methodology. Okay, so this is in the lab. I make my flux. So the way to make flux is just in jar tester. Okay, I created algae blooms water. Okay, and I add aluminum sulfate. Sometimes I use ferric and other coagulants, different conditions. So I make my flux. Okay, so after I make my flux, I want to quantify, okay, how big are the flux? Okay, so I use this very cool machine, master sizer, who can give me can characterize the flux size. But during the PhD, you can have many challenges. And one of my challenges was this instrument only measured spherical flux. But, algae, cyanobacteria is a pain and was giving me a very bad time. During my PhD, I was getting very weird flux. I cannot use a master sizer. Okay? So I went with my supervisor, I got a problem, and she said, okay, let's go to get this. And we got the flock cam. So this is a cool camera from America, and they can take photos and they can measure flock size. Doesn't matter if it's spherical or it's weird things like that. So that saved my day, okay? And also, okay, so I got physical flock properties, I got flock size, but I want to see a little bit what is this making very weird flocks? What is behind that? So I had to do a little bit more fancy instrument. So I use liquid chromatography. And because we like maps, I try to make maps in my flocks, okay? So the thing is to see what is the chemical characterization of my flocks. During the physical flock properties, I can measure flock size, okay? So I make my flocks. What happens is in the treatment plant, they break. You got the filtration process and things like that, they can break. So if they break, they release all the toxins, the cells and things like that. They can grow or not grow. So in the jar tester, I just use different speeds, break my flux, and measure the size. So with that, I get a strength factor, which is a number, and give me like a 0, they cannot regrow, 100%, they can grow after brackets. Okay? So I started to do, okay, what is the flux size of these 4 species plus green algae there? And I get very interesting results. I got two sisters, one sister, the brother, get very similar species, different strains, but the different flock characterization. Okay, and so the flock size was this one was about 600 microns, and this got 113 microns. Okay, so something happened here? They are very similar, but they give me different flock size. Okay, so also I want to see, okay, what happened with the strength factor? Okay, so how strong they are my flocks? So when you see my photos before, the species, I use 2 filamentous species. And these filamentous species, they produce something like that, this kind of flux. Look a little bit like, when you have a long hair and you have a shower, yeah, something like that. So the strength factors say these filamentous species, they got very high strength factor. And the thing I discovered is just they're acting like the backbone theory. So it's something about activated slash. They got like a spine, and they are resistant to break. So this is what's cool thing about this. So that's the strength factor of how resistant the flux to break depending on cell morphology. Okay, and here, okay, so what happened about the flux size? Well, I'm getting similar spherical species, but getting different flux size. So when I go here, I get the concentration of algogenic organic matter. It was a little bit different between microsystem 564 and 555. And the discovery I did during my PhD was the proteins. The proteins was driving the difference between the flux size. Between more proteins, you got a smaller flux. So what is that? When I did the statistical analysis, I see a p-value was relative to a protein content. So what happened is you got proteins, and the protein doesn't like the coagulant. And so, no, they like and consume the coagulant, and that's inhibit to make bigger flux. And also, the proteins and carbohydrates, because it's too much protein, they then link each other, and that's make a little bit this small flux. So when I do this species and the physical properties, and I did the mapping, so here is what I got with the FTIR. So here in this, I got the red thing is proteins, and the proteins didn't like to be with the carbohydrates. And here, when I do the microsystem 564, when a little bit orange is proteins and carbohydrates. So the take message was flux size was driven by AOM, and flux strains, how the flux behave, was driven by cell morphology. And so what does mean for water treatment applications? Okay, so if you got a filamentous species, you're going to have or if you go microsystem 564, you're going to have a weird flux. So maybe you can use sedimentation, or you can use flotation. But it's not very practical from a treatment operator to say, oh, let's go to change from sedimentation to flotation. So, but the message here is it's important to characterize the flux, and important to understand what is inside the flux in order to optimize the process. So for small cells, like making microcystis 555, using flotation or using just filtration process will be enough to remove the flux. Okay? So the main conclusion was that, microcystis different strains can give different flow properties. And filamentous species, they were more driven by the morphology of the species to make the flux properties. In the future, it's recommended to do this. This one is on bench scale. What happened in the real world when you have organic matter, you got different things in the water. So for now, I left here my PhD, but I think it's more room to improvement, more room to research. And thank you to Water Research Australia, the Biomass Group, and my supervisor, Rita Henderson. And yes, that's it. Any questions?
Thanks for the presentation and also for waking everyone up. So do we have any questions for Andrea? There's one there.
Thanks, that was very interesting. So normally when microcystis is grown in culture, it doesn't form colonies. So what might be the implications of the fact that in the real world it's in colonies, and so the flocculation properties might be quite different?
Yes, this is the thing. We have to evaluate that in the real world. When it has the colonies, it was things like that. I think in these ones, it was unicellular cells, but you find colonies, maybe that make a little bit more challenge to the flux to consume the coagulant and things like that. Yes, I think it has to be more research about that, and yes, maybe the flux properties going to be so different.
Another big part, Andrea, you alluded to it at the beginning as well, was actually developing methods that were capable of actually being able to analyse that in the field and testing them on lab-based cultures. So we now have the method and we can then apply it.
I actually want to extend that a little bit further as well. So the other thing is that with the current analysis, we still don't know how much of the AOM. We know that the AOM plays a very big part in the flock properties. So we still don't know if it is 90% responsible or maybe 50% responsible. And so translating that to a real world scenario is going to be completely different. So we definitely have to do more field sampling and field work and running jar tests on field samples to analyze those things in further detail. So it's another question.
Thanks, Andrea. Wish to take a little away from cyanobacteria, but into filtration or flocculation. That's what they're talking about. Jar test, in my view, always a misleading test. For probably flotation, it doesn't matter, but for filtration, it is a poor way of doing it. Because in a real filter, the pores, within the pores, flux are not going to be spherical, as you said. The other thing I'm worried about is the word we use, strong flux or big flux. You don't want massive flux, obviously. You don't want strong flux either, because when you have a strong flux, it'll go and fill the top end of the filter, if you're using filter, if flotation is different. I like to have a bit shearable flux so that they will readjust a bit and they will fill as they go. If you have very strong flux, in my view, it is not the good plan. Why do we look for very strong flux if that's the right forum to discuss about it? Otherwise, we can talk outside. But we don't want very strong flux, in my view. If you are still working on it, I would think we need to look at horses for courses to find out what flux, what strength, and why we need it. Probably I'll leave it there. I mean, I'd like to talk a little more about it and also like to know what you do in this front. Thank you.
Yes, that's one part of the project, to try to see, okay, This one is just making sedimentation, but how we can feed the flux to work in the filtration treatment process, or how can we feed the flux to do the DAF. And I think this is a further work can be done, but I think from a lab thing, this is what we did, the sedimentation, and see what is the flux characteristic, and think more what Rita said about method development, how we can measure flux size and things like that.
The other thing is also that strong, big are all relative terms. So we've also had instances where we've seen filter breakthrough, the flux breaking through the filters and contaminating the effluent. So we need to further define what is strong, what is big, to classify this better.
Yeah.
So any more questions? Okay. Thank you, Andrea. So.